Chromatin immunoprecipitation is a type of immunoprecipitation experimental technique used to investigate the interaction between proteins and DNA in the cell. It aims to determine whether specific proteins are associated with specific genomic regions, such as transcription factors on promoters or other DNA binding sites, and possibly defining cistromes. CHIP also aims to determine the specific location in the genome that various histone modifications are associated with, indicating the target of the histone modifiers. Briefly, the method is as follows. Protein and associated chromatin in a living cells or tissues are temporarily bonded, the DNA-protein complexes are then sheared into 500 BP DNA fragments by sonication. Cross-linked DNA fragments associated with the protein, S, of interest are selectively immunoprecipitated from the cell debris using appropriate protein-specific antibody. The associated DNA fragments are purified and their sequence is determined. These DNA sequences are supposed to be associated with the protein of interest in vivo. Typical CHIP. There are mainly two types of CHIP, primarily differing in the starting chromatin preparation. The first uses reversibly cross-linked chromatin sheared by sonication called cross-linked CHIP. Native CHIP uses native chromatin sheared by micrococcal nucleus digestion. Cross-linked CHIP is mainly suited for mapping the DNA target of transcription factors or other chromatin-associated proteins, and uses reversibly cross-linked chromatin as starting material. The agent for reversible cross-linking could be formaldehyde or UV light. Then the cross-linked chromatin is usually sheared by sonication, providing fragments of 300 to 1000 base pairs in length. Mild formaldehyde cross-linking followed by nucleus digestion has been used to shear the chromatin. Chromatin fragments of 400-500 BP have proven to be suitable for CHIP assays as they cover 2 to 3 nucleosomes. Cell debris in the sheared lysate is then cleared by sedimentation and protein uro-DNA complexes are selectively immunoprecipitated using specific antibodies to the protein, S, of interest. The antibodies are commonly coupled to agarose, cephalose or magnetic beads. The immunoprecipitated complexes are then collected and washed to remove non-specifically bound chromatin. The protein uro-DNA cross-link is reversed and proteins are removed by digestion with proteinase K. An epitope-tagged version of the protein of interest, or in vivo biotinylation, can be used instead of antibodies to the native protein of interest. The DNA associated with the complex is then purified and identified by polymerase chain reaction, microarrays, molecular cloning and sequencing, or direct high-throughput sequencing. Native CHIP is mainly suited for mapping the DNA target of histone modifiers. Generally, native chromatin is used as starting chromatin. As histones wrap around DNA to form nucleosomes, they are naturally linked. Then the chromatin is sheared by micrococcal nucleus digestion, which cuts DNA at the length of the linker, leaving nucleosomes intact and providing DNA fragments of 1 nucleosome to 5 nucleosomes in length. Thereafter, methods similar to XCHIP are used for clearing the cell debris, immunoprecipitating the protein of interest, removing protein from the immunoprecipitated complex, and purifying and analyzing the complex-associated DNA. Comparison of XCHIP and NCHIP. The major advantage for NCHIP is antibody specificity. It is important to note that most antibodies to modified histones are raised against unfixed, synthetic peptide antigens and that the epitopes they need to recognize in the XCHIP may be disrupted or destroyed by formaldehyde cross-linking, particularly as the cross-links are likely to involve lysine amino groups in the N-terminals, disrupting the epitopes. This is likely to explain the consistently low efficiency of XCHIP protocols compared to NCHIP. But XCHIP and NCHIP have different aims and advantages relative to each other. XCHIP is for mapping target sites of transcription factors and other chromatin-associated proteins. NCHIP is for mapping target sites of histone modifiers. Table 1 Advantages and disadvantages of NCHIP and XCHIP, History and New CHIP Methods. In 1984 John T. Lees and David Gilmore, at the time a graduate student in the Lees lab, used UV irradiation to covalently cross-link proteins in contact with neighboring DNA in intact living bacterial cells. Following lysis of cross-linked cells and immunoprecipitation of bacterial RNA polymerase, DNA associated with enriched RNA polymerase was hybridized to probes corresponding to different regions of known genes to determine the in vivo distribution and density of RNA polymerase at these genes. A year later they used the same methodology to study distribution of eukaryotic RNA polymerase II on fruit fly heat shock genes. These reports could perhaps be considered the pioneering work in the field of chromatin immunoprecipitation. XCHIP was further modified and developed by Alexander Warshawski and co-workers, who examined distribution of histone H4 on heat shock genes using formaldehyde cross-linking. This technique was extensively developed and refined thereafter. NCHIP approach was first described by Hebbs al. 1988, and also been developed and refined quickly. The typical chip assay usually take 4-5 days, and require 106-107 cells at least. Now new techniques on chip could be achieved as few as 100 to 1000 cells and complete within 1 day. Carrier chip, this approach could use as few as 100 cells by adding Drosophila cells as carrier chromatin to reduce loss and facilitate precipitation of the target chromatin. However, it demands highly specific primers for detection of the target cell chromatin from the foreign carrier chromatin background, and it takes 2 to 3 days. Fast chip. The fast chip assay reduced the time by shortening 2 steps in a typical chip assay. An ultrasonic bath accelerates the rate of antibody binding to target protein seguro, and thereby reduces immunoprecipitation time. A resin-based DNA isolation procedure reduces the time of cross-link reversal and DNA isolation. However, the FAST protocol is suitable only for large cell samples. Up to 24 sheared chromatin samples can be processed to yield PCR-ready DNA in 5 hours, allowing multiple chromatin factors be probed simultaneously and all looking at genomic events over several time points. Quick and quantitative CHIPA. The assay uses 100,000 cells as starting material and is suitable for up to 1,000 histone chips or 100 transcription factor chips. Thus many chromatin samples can be prepared in parallel and stored, and Q2 chip can be undertaken in a day. Microchip. Chromatin is usually prepared from 1,000 cells and up to 8 chips can be done in parallel without carriers. The assay can also start with 100 cells, but only suit for 1 chip. It can also use small tissue biopsies and microchip can be done within 1 day. Matrix chip. This is a microplate-based chip assay with increased throughput and simplified the procedure. All steps are done in microplate wells without sample transfers, enabling a potential for automation. It enables 96 chip assays for histone and various DNA-bound proteins in a single day. Chip has also been applied for genome-wide analysis by combining with microarray technology or second-generation DNA sequencing technology. Chip can also combine with paired end-tag sequencing in chromatin interaction analysis using paired end-tag sequencing, a technique developed for large-scale, de novo analysis of higher-order chromatin structures. Limitations of chip. Large-scale assays using chip is challenging using intact model organisms. This is because antibodies have to be generated for each TF, or, alternatively, transgenic model organisms expressing epitope-tagged TFs need to be produced. Researchers studying differential gene expression patterns in small organisms also face problems as genes expressed at low levels, in a small number of cells, in narrow time window. Chip experiments cannot discriminate between different TF isoforms. See also, RIP chip, a similar technique to analyze RNA-protein interactions, DAMID, an alternative location mapping technique that does not require specific antibodies, ChipExo, a technique that adds exonuclease treatment to the chip process to obtain up to single base pair resolution of binding sites. Chip-on-chip, combines chip with microarray technology. References. External links. Chromatin Immunoprecipitation at the U.S. National Library of Medicine. Medical subject headings, epigenome.noe.com, chromatin immunoprecipitation on unfixed chromatin from cells and tissues to analyze histone modifications, chromatin immunoprecipitation of protein complexes, mapping of genomic targets of nuclear proteins in cultured cells.